Okay, it's another quick tour through the tissues. We've got, they've divided this between isolating connective tissue cells, which to tell you the truth, I've never seen that term before. Defensive connective tissue cells, I dug this overhead out of a big pile of overheads that I had. Structural connective tissue cells, again, what connects them is the setup, right? Floating in plasma. Well, I can't tell you, it's such a fat inside the cells. It's never made any sense to me. We've got nervous tissue, which is obviously all through the body, but it's concentrated in the central nervous system. Muscle, cardiac, skeletal, smooth, epithelial. Here they're showing the three shapes: squamous, columnar, tall skinny ones, and cuboidal, these kind of squarish ones, very often wrapped around little projections. A couple of other things that have to do with animal structure. Whoops. Oh, goodness. Body spaces. Early animals, multicellular systems, would have had spaces that are technically external spaces. Is that the spaces around the animal and the spaces inside the endoderm, inside the digestive system, that are connected to the outside, they never actually are inside the body. The spaces in there connect to the outside. And in early systems, the cells that differentiated into different types of organs and such, that's with the introduction of mesoderm, still all stayed connected to one another, except for spaces that connected to the outside. The introduction of internal isolated body spaces, what they call, well, I don't want to give you the terms because the terms get a little dicey. But the internal body spaces where the organs can kind of hang in a liquid-filled space is a very important evolutionary development. Think about this. You have your heart going dun, 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 next to your lungs going, ugh. So they're going at very different rates. If they were connected to one another by a cellular continuum, they would be interfering with each other's movement much more than they do hanging in the space, surrounded by slippery membranes, so they basically are just, they're not really rubbing on each other. That's why if you get like a heart infection, if it spreads to the surrounding membrane, the pericardium, and interferes with its sliminess, it can literally, you can wear a hole in your lungs with your heartbeat. But they can, under normal conditions, move independent of one another. Your intestines and your stomach and your small intestine, your large intestine, your stomach, all contract at different rates and are surrounded by other things that aren't contracting at all, and things that need to contract on a controlled system like your gallbladder. But they can all do that because they're hanging in a space semi-independent of one another. So body spaces becomes a really big deal in the evolutionary development of animals. These were originally called homeotic genes. Then they, for a while, called them homeobox genes. Then they've gone to Hox genes. I think right at the moment when you see them, it tends to be capital H and little OX. For a while, it was all caps. So this is a term that you'll see if you look at older and older things going back maybe 30 years. You'll see different versions of the same concept. And I should have covered it earlier, actually, now that I'm thinking about it. But this is where it is in my notes. So the cells are, early on, you got a zygote, you got embryo cells. They're just in the business of making lots of cells. It's too early to get the cells to start to do different things. Then you get the layers. You kind of have the setup of the beginnings of your different organ systems. And now it's time to start getting different parts of the body to do different things. That's what activates the Hox genes. Your Hox genes are your basic early layout system genes, where there's Hox genes that go basically, okay, this is gonna be the front end of the animal, this is gonna be the rear end, anterior, posterior. In bilaterally symmetrical animals that are not completely symmetrical, and they pretty much never are, we have Hox genes that determine the liver's gonna wind up on the right side, and the heart's gonna shift to the left side, and your stomach's gonna be on the left side. They all start in the middle, but there is a genetic initiation that moves stuff asymmetrically. It tells appendages where to grow. It lays out the skeletal system. It somewhat involved in the beginnings of the nervous system. These are basic layout genes. In some cases, in things like that are well segmented, they actually are laid out on chromosomes in the order of the, like, you know, if you got a gene and a gene and a gene and a gene and a gene. I guess I should go this way. This screen is backwards for me. Then you'd have like a head activator and then a thorax activator and an abdomen activator. It actually lined up on the chromosomes that way. These are ancient genes. They're very similar across a broad swath of different animals. Okay, this is a fairly short video anyway, so I'm going to tell you the story. If you look at insects and vertebrates, provided you don't look very closely, but insects and a lot of other arthropods have a multi-lens vision system, what they call compound eyes. Lots and lots of lenses, little clusters of receptors behind each lens, and that's how they see the world. We have single lens eyes, big old lens, good old space, and then a screen with a bunch of receptor cells on it. Pretty different setups. Turns out arthropods have single lens eyes too, but because of the differences, people who weren't paying a lot of attention considered that down the family tree, wherever our branch and the arthropod branch met, those animals didn't have what we would consider eyes. They might have been able to detect light, but they didn't have specialized vision structures that we would consider to be eyes. And so it's not an ancestral trait. It's a derived trait. It's that compound eyes going up to the arthropods, single lens eyes going up to the vertebrates. A fruit fly mutation causes the Hox gene for eye location to malfunction, and it puts these little lenses all over the place, on their butts, on their legs, on their wings. And so they went, oh, this is a basic eye layout gene. We're not expecting it to have an equivalent in vertebrates because the eyes evolve separately on the different branches. But let's go look anyway. Let's take the sequence of this gene and try to match it to a sequence in mouse genetics. And they found that sequence. Now there is something we'll talk about the second semester where under the right situations, you can actually get into and develop a strain of mice where you completely deactivate a particular gene. That's a way to see what it does. And you produce a strain called knockout mice. You've knocked out that particular gene. Now, a lot of genes you can't do that because it just kills them, but some of them you can knock it out and see what happens. Well, they produced a knockout for this Hox gene in mice, and you don't get a mouse with little eyes all over the place, but you do get a mouse with no eyes. They have a dent in their skull, but no eyes develop there. So that's a pretty strong indication that our shared ancestors were already building eyes on a basic layout system. And one of them went up to the compound eyes and one of them went up to the single lens eyes. And that was one of the earliest Hox genes that got a lot of attention. And since then, they've looked at a lot of stuff. Another big deal with animals are what are called symmetry patterns. These are very connected to different groups. Usually a symmetry pattern fits an entire group, very often a whole phylum. I'm going to write these down and then talk about them. Okay. I was gonna step back out here and talk to the screen, except I've got something in my eye, so I'm gonna stand over here and rub my eye while I talk about this. Radial symmetry is a circular pattern where the animal is on one axis is round, not spherical. Spherical is a type of symmetry, but you don't typically see that in animals. You do see it in protozoans, but not in animals. So it tends to be a column, and if you take a pie slice, you're looking down at the column, you take a pie slice, then what's in that pie slice would be equivalent to the same pie slice taken another way. It's symmetrical, but it's a radially symmetrical. And sometimes it is a particular numeric radial symmetry. The group that has starfish in it are pentaradially symmetrical. So if you take a pie slice that is a fifth pie slice, then what's in there is going to be in any other pie slice. If you get a whole starfish arm, you'd have a whole starfish arm, even if your slice had a half and a half. And the other things inside the animal are also pentaradially symmetrical. Sea anemones are octaradially symmetrical. Sometimes a lot of cnidarians are quadraradially symmetrical. So there could be numbers, sometimes not. Hydras, for those of you that saw the hydra in the classification lab, have varied numbers of tentacles, and internally they don't have a numeric radial symmetry, but they do have a radial symmetry. A pie slice is just not a particular size pie slice that's equivalent to the other pie slices. Bilateral, two sides. Right side, a reflection of the left side. So we are bilaterally symmetrical. It is a very common type of symmetry in larger animals. And in our distant ancestors, when you have two sides, then, and you're moving through the water, because our distant ancestors moved in the water, right? Two sides driving you. That tends to produce a front end and a back end, because efficient movement tends to focus on, maybe you can back up if you need to, but typically you're better off going in a particular direction. That means you meet the world with one end of your setup. It'd probably be a good idea if you could read what was in front of you in terms of good things and bad things. And so what you find, cephalization, in bilateral animals, there tends to be a specialized front end of both sensors and processors. Cephalization means development of a head. It's not always a head, but there's a head-like structure at the front end where you're tasting, you're smelling, you're seeing, in some cases, what you're about to run into, so that maybe you can avoid it, or maybe go toward it if it's good, avoid it if it's bad. And a little bit of that on the back end for things that sneak up on you. But it does tend to be a front-back specialization in the nervous system. And it produces, brains are the equivalent of brains. So it's in bilateral animals, they tend to have a front end that's highly processor-oriented, and a back end that's pretty highly process-oriented. And then a lot of stuff driving on the sides. Okay, that is it for animal structure. But we're not done. We're going to go through the major animal groups so that all this stuff I've been talking about, you'll have a little bit better background when I start to apply this about this system to an animal group, or this about this system to another animal group. It'll make more sense.